It's my pleasure now to introduce Tomasz Wilmanski, who's going to talk to us about defining a healthy gut microbiome. There's actually a question a little bit earlier about disease versus healthy gut. He's going to talk about insights from analyzing deeply phenotyped human cohorts. Tomasz, you're welcome to. You're on screen. We can hear you, or if you want to share your screen.
Great, thank you. Thank you for the introduction, Len. Let's see if you can see my slide as well.
You are all good to go.
Okay, great. Well, thank you. And I've really been enjoying the talks today, the ones I was able to catch earlier. So my name is Tomasz Wilmanski, and I'm a research scientist in the Hood Price Lab group for systems biomedicine, located in the Institute for Systems Biology in Seattle. Our two top PIs are Lee Hood and Nathan Price, and some of you may have heard Nathan talk about some of my work last year at the same symposium. But we're a big group of researchers studying various aspects of biology, and these are our main research areas. The ones highlighted in orange are the ones that I'm really focusing on: is longevity and aging, scientific wellness, and this concept of PE3 cloud, or personal dense dynamic data cloud, which I'll discuss in more detail. And gut microbiome research is kind of on the forefront in all of these areas. And that's where my expertise really lies in the lab. So I'll jump into a question or a challenge that has time and time again kind of challenged the field in general. And it's this concept of what is a reference or a healthy gut microbiome? One example I like to use is from a recent 2018 Nature Metabolism Medicine paper. So this is a map of Guangdong province. This study in particular sampled 7,000 individuals across 14 districts in the province. The first thing that might pop out is the bar plot showing the relative abundance of phyla for each individual in the study. And you can see, looking at Firmicutes, Bacteroidetes, the major phyla, there's a huge swath of different microbiome combinations and compositions across the different districts. What's particularly interesting about this study is when you try to train a machine learning algorithm to, for example, distinguish type 2 diabetics from healthy or non-diabetic individuals, you can do pretty well when you do it within a district. And that's shown on the right on the blue points when you do a tenfold cross-validation within a district. The moment you use the same model to predict type 2 diabetes in another district or across the whole province, the performance really drops. And you have the same thing for metabolic syndrome and a number of other conditions that the researchers tested in this study. The conclusion that they posted is that localized baseline and disease models need to be built in order to predict metabolic risks. But another conclusion that can be drawn from that is that there's multiple taxonomic combinations within the gut microbiome that can be considered healthy. It's very hard to define certain specific taxa that can always contribute to type 2 diabetes, although we are getting close at that. Sometimes higher level measures seem to be a bit more robust as markers of microbiome health, although they may be less specific. I'm sure everyone's familiar with alpha diversity, or this within sample diversity measure. How diverse is your ecosystem? And there's been a ton of work showing that when people take antibiotics or when they suffer from obesity, alpha diversity is generally lower. When you have a foodborne infection or diarrhea, alpha diversity drops even further. And certain individuals with very low alpha diversity are higher risk at reoccurrent C. difficile. On the other spectrum, there's been several studies showing that hunter-gatherers or non-industrialized populations tend to have higher alpha diversity. And we think of these individuals as being healthier, having healthier diets, less processed foods. And this concept that higher alpha diversity you have, the healthier you are. But at the higher end of the spectrum, we also have certain individuals who suffer from constipation, for example. Constipation has been associated with higher alpha diversity, which makes this idea that maybe there's a Goldilocks zone in terms of alpha diversity, but also makes this metric an interesting starting point in our pursuit of defining a healthy microbiome. And the way we really approach it is kind of as an alternative to focusing on the microbiome, but more as a complementary approach, is look at the gut microbiome within the rich phenotypic context of the host. So we've been generating what we call personal dense dynamic data clouds for thousands of individuals, where we combine blood metabolomics, proteomics, clinical labs with a panel of self-reported gastrointestinal health and disease metrics on top of 16S ribosomal RNA gene sequencing data, and as well as human genome. And once we start integrating all of these data types together, we start to see a better picture of what we can consider a healthy microbiome in many ways. So the first study that really pioneered this was a study from our lab called the Pioneer 100 study. We took 108 volunteers and performed deep phenotyping from all the omics I discussed earlier every 3 months, and identified correlations and certain markers of disease transitions in metabolic health and genetic reflection of genetic risk as well. This Pioneer 100 study kind of transitioned into generating a startup company called Aravale. It was a consumer-facing personalized wellness platform that operated from 2015 to 2019. In that process, we were able to enroll over 5,000 subscribers. We measured over 1,000 analytes in the blood every 6 months, combined it with all the other omics platforms I discussed earlier. On top of that, we also had lifestyle coaching. But this rich wealth of data really allowed us to tackle this microbiome health question. So we combined different vendors, as you can see. For microbiome sequencing, we used Olink for plasma proteomics, Metabolon for our metabolomics analysis, and the big clinical lab vendors in the US, LabCorp and Quest Diagnostics. So initially, out of these several thousand people we had at the time, we specified very strict criteria. If we want to kind of see how gut microbiome health is reflected in the host, we wanted a few specific kind of check marks to be kind of fulfilled by individuals. So first, we specified that a blood draw for analysis had to be taken within 21 days of a stool sample. Initially, we just used a single clinical lab vendor to avoid any batch effects or any differences between lab vendors in measuring the same lab, like LDL cholesterol, HDL cholesterol. Interestingly, we also had because of how we process the samples, how we collect the samples, we were able to compare metabolomics, proteomics, and clinical laboratory tests, all taken from the same fasting blood draw, which really gives us a nice comparison how these different platforms reflect different aspects of the gut microbiome. We also didn't do a lot of exclusion. I know there's a lot of discussion here to match individuals in case control studies or to avoid certain confounding variables like antibiotics use. But we actually wanted to see how the host phenotype is reflected per terms and how resilient it is to an antibiotics exposure. Do the same relationships between the gut microbiome and the omics hold when you take antibiotics versus when you're not on antibiotics? And then we validated our findings from these 400 individuals in an additional validation data set. And these were individuals who enrolled in the program later. And we switched microbiome vendors. So they also used a different microbiome vendor for their microbiome analysis, which we believe that if we can validate our findings with these differences, that means that these findings are pretty robust. A real comment about the cohort itself. Majority of the individuals who subscribed to Aravale were residents of California and Washington. And although on average, they tend to be a bit leaner, there's less prevalence of obesity in our cohort. A lot of the lipid markers are quite similar to the global national averages in the United States. And the cohort is very representative of the populations that live in Washington and California. So our initial test was really to look at metabolomics or plasma metabolomics. And the question is why focus on the blood metabolome? And when we performed the study, there's been several really nice mouse models and animal studies showing that there's this intimate relationship between metabolites in the blood and what the gut microbiome actually produces. This is from a 2009 PNAS paper by Wyckoff et al., showing that if you measure the metabolome of wild-type mice and then germ-free mice, there's a number of different metabolites that fall below threshold of detection in the blood. And as an example, this is just tryptophan metabolism, which many of you may be familiar. Gut bacteria can break down tryptophan into indole, and then there's several different indole propionate, indole acetate, which eventually enters circulation. Indole itself enters circulation, but is further metabolized in the liver into indoxyl sulfate. But in many cases, and it's shown in the box plots at the bottom of the slide, In germ-free mice, many of these metabolites, even though they're fed the same diet, are almost either below threshold of detection or very low or decreased. And in addition, tryptophan, which is a substrate for microbial metabolism, is actually increased in the blood, showing that gut microbes can metabolize a bunch of metabolites, but also that these metabolites end up in circulation and can affect host physiology. So we focused on machine learning based approaches, and we tried several, but eventually focused on what's the most interpretable and easiest to understand, which was a penalized linear regression model. So it's a linear combination of features. And then we did a tenfold cross-validation scheme where you train the data on 90% of your cohort, and then you predict the 10% that your model has never seen. And you can do this 10 times for each of these 10, this 1/10 fraction of your cohort, and you get a more conservative estimate of your prediction. And in short, we were surprised that we could actually do a pretty good job at predicting individuals' alpha diversity using just a blood metabolomics panel. Not only that, we were able to predict alpha diversity using just 40 metabolites from the 659 measured by Metabolon. Of those 40, there were actually 11 core metabolites that appeared time and time again as being the most influential in predicting alpha diversity across all 10 cross-validations. And depending on the metrics, since there's several alpha diversity metrics, Shannon, Chow, and observed species, we're up to 50% of alpha diversity variance could be explained by a very small metabolite panel, which is really exciting. When I mentioned earlier that we didn't really have exclusion criteria, and that one of the reasons was we wanted to see if the metabolome actually reflects disease perturbations. We didn't want to kind of eliminate diseases. We actually wanted to include them and see how the metabolites changed as different diseases emerge. So in this case, what you see here is a beta coefficient or effect size from models predicting either Shannon diversity, what we observe and measure from 16S RNA sequencing, or this M Shannon, which is a metabolome prediction of Shannon diversity, which you could theoretically calculate without ever sequencing the gut microbiome, just looking at the blood. And what we see it's highlighted in red, the major disease conditions where we see this shift, but with abdominal pain, diarrhea, type 2 diabetes, whenever observed alpha diversity goes down, and what's been shown in previous studies, or goes up, the metabolome reflection actually follows very closely. So people with type 2 diabetes, their metabolomic reflection of alpha diversity decreases just like their observed alpha diversity. In addition, we really want to tackle this antibiotics question. Do antibiotics disrupt these metabolite microbiome relationships? And we did this by comparing the prediction of our algorithm, looking at either the whole cohort, that's the gray bar to the right, or non-antibiotics users who answered the antibiotics questionnaire and clearly stated they have not taken antibiotics. And also and using both the 40 metabolite model in red that we created or the whole metabolome. And the conclusion from this, what I really want to emphasize is that whether we exclude antibiotics users, we include antibiotics users, the prediction is just as robust, meaning that antibiotics impact the gut microbiome, but the metabolites we measure capture that perturbation quite well. There's no drop in performance in our algorithm. In fact, the only time we actually saw a drop in performance in how well we can predict alpha diversity using blood metabolites was when the time lag between the blood collection and gut microbiome sampling or stool collection increased. So this is just a bar plot showing the performance of our algorithm, but the individuals were split into tertiles, tertile 3 being your blood sample was taken approximately 2 to 3 weeks after or before your gut microbiome sample. And even then, though there was a decrease in performance, we only explained 30% of the variance in individuals who had this big time lag. We still captured about 30% of the variance, meaning that this blood sample can give you a pretty good reflection of your microbiome state within a 3-week window, which is quite exciting. What I think further really emphasizes the metabolome as this interface between the gut microbiome and the host is that we validated our model in the separate validation set where a different microbiome vendor was used. And you can see the performance was quite similar. We explained about 34% on this additional data set while the whole metabolome, not the 40 metabolite model, explained marginally more percent of the variance. But what's really interesting is that clinical labs in both the discovery and validation cohort explained a very small proportion of the variance in alpha diversity, which is particularly striking because clinical labs is what we rely on to assess health in the clinic, and they seem to capture very little of what might be going on in the gut microbiome. So I've been talking a lot about this model, but what are actually the metabolites that predict your alpha diversity? And many of the top hits is what you might expect. They're microbial or human microbial co-metabolites. They're metabolite generated by the microbiome, like precressyl or benzoate or cinnamate. And then they're further conjugated in the liver into cinnamyl glycine, precressyl sulfate, and hippurate. And those were three of some of the strongest positive predictors of alpha diversity. There were several secondary bile acids like arsalucodeoxycholate. Interestingly, the strongest or most influential predictor of alpha diversity across both cohorts and all the 10 cross validations was a sex steroid hormone, 5-alpha androsten 3-beta 17-alpha diol sulfate, which is a breakdown product of testosterone. And it's actually quite consistent with some animal studies showing this intimate relationship between sex steroid hormones and gut microbiome composition and metabolism. But there was also a number of metabolites on the host side. There was also PFAS, which is perfluorooctanosulfic acid, which is actually a contaminant, or it's a chemical compound found in different 3M products. And that was positively associated with gut alpha diversity, to our surprise. What's further interesting is that a lot of the metabolites we identified as these predictors are actually bioactive compounds that have been shown to influence host physiology. Many of you may be familiar with TMAO, which has been shown to promote cardiovascular disease. And that was actually a positive predictor of alpha diversity. The higher your TMAO is, the higher your alpha diversity. Indole is one of a tryptophan breakdown product, and it's been shown to have antioxidant capacity, certain neuroprotective properties. Hippurate mostly acts on the liver, but actually has been shown to help mitigate or reverse metabolic syndrome. Precressyl sulfate is potential uremic toxin that's also has been a positive predictor of alpha diversity in our models. With these metabolites and the gut microbiome kind of combining all these omics together, we can start seeing more of a picture of this Goldilocks zone in terms of gut microbial alpha diversity. There's clearly certain metabolites in the blood that decrease as your alpha diversity goes down, like imidazole propionate, which is known to promote insulin resistance. There's many beneficial metabolites that increase as your alpha diversity goes up. But some of them, like TMAO, are actually mildly toxic or can be toxic at higher concentrations, similar to precressyl sulfate. So this Goldilocks zone emerges where, yes, high alpha diversity is beneficial, but too high alpha diversity actually might be detrimental. Some of the ways we think about how metabolites could be incorporated moving forward is they're a nice readout, a functional readout of the gut microbiome. 16S metagenomics gives you an idea of what's there, what genes are present, what enzymes are present. But the metabolites in the blood are actually the functional outcome, the combination of your diet, your microbiome composition. What is that, the ultimate product that can affect your physiology? It flows in the blood. It can target different tissues. So it's a very strong interface that we should focus on more. We also believe it's a potential initial screening in a clinical setting. It doesn't give you the full picture of all the different aspects of gut microbiome health, but it could be the initial starting point in a clinic from a blood draw where this indicates your alpha diversity or microbiome composition may be altered or maybe something may be going on. Let's sequence your gut microbiome to figure out what exactly is going on. I touched upon this earlier, but clinical laboratory tests seem to capture a very small percent of variation in gut microbiome structure, indicating we might need different tests or complementary microbiome tests to give clinicians the information they need to kind of get a full picture of one's health. And the other aspect we're still focusing on is how many more unique microbial metabolites are there in circulation that we don't really know about, and what is their biological impact on host physiology? I think that's a very interesting and exciting question moving forward. The last two slides, I just wanted to explain how, from a biology and nutrition perspective, how we think about this. My background is in nutrition science, so this is very close and dear to me. But how can such a small number of metabolites explain such a complex system to some extent? And I think the idea is partially in what we consume and how we consume it. If you look at macronutrients, micronutrients, the essential food compounds that we consume in our diet, amino acids, fatty acids, vitamins, minerals, they actually comprise a very small proportion of the diversity of what we consume in a diet. And many of them are actually absorbed to a large extent in the small intestine, where microbial communities are less abundant than the large intestine. What actually ends up in the large intestine is, as we talked about earlier in many talks, is fermentable fiber, but also this very diverse group of phytochemicals. Over 8,000 structurally different polyphenols have been identified, and over 1,000 have been quantified in foods. So this diverse group of phenolic compounds and other phytochemicals enters the large intestine. They have known microbiome modulatory properties. Many of these phytochemicals in plants are actually serve a purpose kind of to be antimicrobial. So they can actually impact your microbiome composition. But the gut microbiome is also capable of metabolizing these different phytochemicals into smaller molecular weight compounds that are more universal. Hippurate, hydrocinamate, or hydroxyphenyl acetate actually break down products of very complex polyphenols that once you break them down, many polyphenols can give rise to the same breakdown product. And maybe this is the idea behind how such a diverse system with such a diverse dietary input can be monitored or interpreted to some extent from a much smaller number of metabolites in the blood. And we're moving forward. We think we have some evidence for this, but I think it's not a fully answered question. With that, I would like to sincerely thank my whole research team, but particularly Noah Rappaport, shown here in the red box, who was the co-first author on this paper, as well as Sean Gibbons, who's a close collaborator and a big microbiome researcher, great colleague as well. Thank you so much. Really appreciate having the opportunity to talk.
Tomasz, it's an excellent presentation and food for a lot of thought. There's a couple of clarifications. You talked about the applicability in a clinical setting. One of the questions that's come in is what's the sample size? Can that theoretically be even broader than a clinical setting? Do you think this could emerge as an in-home type of environment as we move to telehealth, that type of system?
Yeah, I think so. I mean, that's what I'm really excited about, because if you think about these metabolites and how dynamic they could potentially be, you could start thinking of these N-on-1 personalized approaches where you measure your microbial metabolites in the blood, and then you change your diet and see how they change a week from now or 2 weeks from now. And I think that's a great way to personalize your own health on like an N-on-1 approach.
Yeah, just specifically, what's the blood sample size? What amount are we talking? Is it a little pinprick or is it a draw?
So metabolomics, for their broad untargeted metabolomics panel, uses a very small amount of blood. We take a clinical lab blood draw. Majority of that blood goes to measuring your LDL, HDL. A tiny fraction of that is sent to Metabolon for, my memory tells me it's around 100 microliters, but I'm not exactly sure about the number. All I know, it's a very small volume compared to what you need for clinical blood tests.
Got it. You had some. You showed the robustness of the system to withstand the antibiotic environment. You want to go through that a little bit and what some of the implications, and are the metabolites adjusted or do they differ once you understand? Like, is there any change that happens with the antibiotic environment?
Yeah, so we really want to ask that question because if you have this great model, but the moment someone takes antibiotics or has some disease, the model falls apart, it's not a very useful model. What we try to show is that antibiotics will decrease your alpha diversity, but all the metabolites that are associated with alpha diversity move in the same direction, it seems like. So the metabolites are able to capture antibiotic perturbations to some extent. It's a bit of a murky reflection in the blood. We don't get all the intricate details you would get from metagenomics, but we do capture antibiotic perturbations to some degree.
Okay. There's a specific question about insulin resistance and propionates. Maybe you could sort of dig into that just a little bit.
Yeah, so propionate in general, the short chain fatty acid, or is it, okay, yeah. So I think short chain fatty acids in general, butyrate, and those big one, propionate as well, can help with insulin resistance. Type 2 diabetics often have lower capacity to produce short chain fatty acids in the gut microbiome. there's a number of mechanisms that have tied short-chain fatty acids to insulin resistance, and prebiotics as well, which kind of are the substrate for propionate production.
Great. Okay, some of the metabolites, you referred to TMAO as a signpost of alpha diversity is too high. Could this be related to meat protein intake? You also referred to one of the metabolites that was involved in the testosterone pathway. Would that vary, and how do you account for sort of gender differences? So a couple of those questions on the specificity and how that then back correlates to either gender or diet.
Right. So we had some, but I'll tackle the gender question first, and then I'll move on to the diet one. Since our strongest predictor of alpha diversity was a sex steroid hormone, we did a lot of sex stratified analysis to see if, like, is this mostly in men or women? And what you see is that when you plot this on the graph, men have more of this metabolite than women, but relative to alpha diversity, they both have parallel lines of a positive relationship. So even though there are sex differences, the relationship with your gut alpha diversity is almost exactly the same in men and women, which further speaks to the robustness of the model. With diet, we had a pretty crude estimate of people's dietary intake from our cohort. People could self-report whether they were on a paleo diet or on a vegan diet. And there were some differences in alpha diversity based on the diet. But the metabolomics model that we created captured all those differences really well. So vegans tend to have a bit higher alpha diversity. The metabolomics prediction captured that in vegans pretty well. So it seems like across diets, it works relatively well.
Excellent. All right, we are out of time, but I want to thank you for your presentation. I think this healthy gut microbiome and actually making it a practical reality in our day-to-day lives and measurements and getting it into the health equation is extremely important as we move to the future. So thank you, Tomasz.